All right, and welcome again, everybody, to our 8th anatomy tutoring session. I want to remind everyone that this is the final tutoring session for 2024, so we'll pick back up again in the new year after today. That'll occur on January 5th, and that will actually be our final tutoring session for the local competition before our review sessions. And I know we've had a lot of good questions about what those review sessions are going to entail, and just want to give you guys a heads up that if you really enjoyed our interactive Q&As that we're doing, that our lovely presenters have been doing throughout your tutoring sessions, you can expect that your local review session is going to be held very similarly to that, where it's going to be all content derived for questions that could be on that examination, and it'll be very interactive. So instead of having sort of content that we'll review, it'll be driven again by us polling you guys with questions. You can assess where you're at and get a feel for what questions are going to be on that local competition exam. I wanted to remind everyone that the signups are due by the 13th of January by 12 midnight Eastern time. So if you have any peers or any friends that were wanting to sign up, remind them that that deadline is very quickly approaching. And then the final thing that I wanted to say, just as a reminder, is we have our research survey. Again, it's completely optional and it doesn't affect your standings within the competition whatsoever. But if you're interested in seeing what that research was like, and you're interested in helping us find out that information about your motivations and what you're interested in participating in STEM, then I'll be dropping that link in the chat again as well. So last thing I want to say before we get started is I hope everyone's having a wonderful start to their holiday season. I see Dr. Frank is as festive as ever and ready to moderate our session, so I'll turn things over to him to get us started.
Ho, ho, ho. Welcome, everyone. Tonight we're going to be talking about the urinary and reproductive systems, and our first presenter is Drexel's very own Zach Mattiola. Okay, Zach, if you've been to many of these sessions, Zach is one of our pros. At this point, I think this is third or fourth at least. So Zach is from Collegeville, Pennsylvania. He graduated from the University of Michigan in 2022 with a degree in neuroscience and is currently a second-year medical student at Drexel University. In his free time, he likes to watch sports, especially Michigan football, the Eagles, and the Sixers. He also likes to work out and play basketball and pickleball. Academically, he is interested in pharmacology, anatomy, and physiology, and he wants to specialize in anesthesiology, critical care medicine, or surgery. So with that, Zach, the floor is yours.
Great. Thank you, Dr. Frank. All right. Let me just share my screen here. All right. Nice to see you all again. Thanks for joining us on this Sunday night to learn about the urinary and reproductive system. So we're going to kick it off with some anatomy here. And here are our learning objectives for today. So we're going to learn about the urinary system, the kidney, all the way down to the external urethra and all the anatomy in between. We'll look at the differences between the male and female urinary systems, and then we'll get into the male and female reproductive systems. And just as a disclaimer, so when I'm talking about male and female during this lecture, I'm referring to biological sex, not gender. Male is going to be referring to testosterone-driven anatomy, and females are referring to estrogen-driven anatomy, just as a heads up, okay? All right. So the first thing we're going to talk about is the urinary system. So the start of the urinary system is the kidneys, okay? And the function of the kidney is going to be filtering blood to remove any waste or toxins or unwanted substances that your body wants to get rid of. So anatomically, you have two kidneys, okay? And the right kidney is going to sit lower than the left kidney. And the reason for this is you have your liver sits right above the left kidney right here, and it's going to take up a lot of space. So your right kidney is going to sit lower. Okay, so your kidney is going to remove toxins from blood. It's going to get blood from the renal artery coming off the aorta, and then it's going to filter that blood, and it's going to send that filtered product into a structure called the ureter. And each kidney has a ureter. It's going to send what it filters into the ureter, and the ureter is going to carry that filtered fluid down all the way to the bladder, and then you're going to urinate. That's how urine is made, and you're going to urinate that out. Okay. So the bladder has, so now we've gone from the kidney, we're now in the bladder. Okay, the bladder has a wall which is kind of composed of this folded tissue called rugae. And these rugae, when the bladder starts to expand, the rugae start to flatten out and allow the bladder to get real full with urine. And I think you've learned about the stomach already. The stomach kind of has a similar system built in. It has all these folds in it, which it allows allowing it to expand to accommodate any extra volume. And then we have a layer of smooth muscle also surrounding the bladder called the detrusor muscle. So when it's time to urinate, that muscle is going to contract and it's going to squeeze the bladder and allow for urination to occur. Okay. And then you have the urethra. So here is the male anatomy. You see the urethra right here, this kind of tube that connects the bladder to the outside world. Okay. And this is what the urine is going to pass through in order for urination to happen. Okay. And then here's the female bladder and the female urethra right here. Okay. Now, so the differences between the male and female bladder. So the male bladder is going to sit in front of the rectum. So this is the rectum right here. Okay. The male bladder sits in front of the rectum and above the prostate gland, which is right here. Okay. Now, the female anatomy internally is a little different because they have different sex organs. So the female bladder is going to be located in front of the vagina, which is right here. Okay. And here's the bladder, the female bladder, and it's going to be below the uterus, which is right here. Okay. So in the male version, you don't have anything between the bladder and the rectum, but in females, they have the vagina and the uterus sitting between the bladder and the rectum. Okay. So now we're going to look at each individual sex by itself now. So for the female urethra is real simple, okay? It's very short. It's about 4 centimeters long, and you know, it just extends from the opening or the bladder to the outside world. It's very short, very simple. Okay. Importantly, you have two structures called the external and the internal sphincter. Okay, the internal sphincter is essentially at the opening of the urethra, and then the external sphincter sits right before the opening of the urethra to the outside. Okay, and this just allows you to control urination, right? You don't want to, if you didn't have sphincters there, you would just have fluid pouring out of your bladder constantly. So you have a sphincter that can open up and close up to allow control of urination. Okay. Now the male bladder is a little bit more complicated. Okay. The male bladder is very long and it's composed of 3 sections. And importantly, in the female bladder, only urine is going to pass through the urethra, but in the male system, you're going to have urine and semen passing through the urethra. Okay, so the urethra in males are composed of 3 different sections. Okay, the first part is called the prostatic urethra. Okay, and it's named that because it's passing through the prostate gland, right? Okay, then you have the membranous urethra, which is a very, very short part of the urethra right here. And then finally, you have the spongy urethra, which is the longest portion, and it's called the spongy urethra because it's passing through the penis, and there's a part, it's passing through what's known as the corpus spongiosum. So it's called the spongy urethra. Okay, so we have 3 parts: prostatic urethra, membranous urethra, and spongy urethra. Okay.
Hey, Zach,
All right, now.
Dr. Peterson, maybe before you move on, just go back and explain the difference between the types of muscles for those two sphincters so they better
Yeah,
understand.
that's,
One is
yeah,
under involuntary control, and the other
absolutely.
voluntary.
Yes, so the internal urethral sphincter is composed of smooth muscle, and that is involuntarily controlled. Remember, smooth muscle, you can't consciously contract or relax smooth muscle. The external urethral sphincter is skeletal muscle. So you can decide to relax the external urethral sphincter to urinate, because you can control when you want to urinate and when you don't want to urinate. And that's this external urethral sphincter, which is skeletal muscle, allows you to have voluntary conscious control over urination. Okay. All right, now we have a quick question for you to answer. So which structure in the bladder allows the bladder to expand and accommodate large volumes of urine? Got a little bit of a horse race going on here.
All right, get your final answers in. If you don't know, go ahead and guess and that'll give us a sense for where you're at so far. Go ahead and close the poll in 5, 4, 3, 2, 1.
All right, good. So the answer is, in fact, rugae. Okay, so I guess the rugae are essentially just like folds of tissue. Okay, so think about like a plastic bag that's all wrinkled up and crushed up. When you pull that plastic bag apart, those wrinkles are going to flatten out, and that's what allows the bag to get bigger. So the same is true here. Okay, you have a lot of folds in here. You see all these folds, and as the bladder fills, those folds are going to flatten out and allow for extra surface area. Okay. All right, another question here. So why does the right kidney sit lower than the left kidney? Good. Seems like you guys got this one down.
All right, we'll go ahead and close this poll in 5, 4, 3, 2, 1.
All right, good. Exactly right. So it sits lower to accommodate the liver. So right above that right kidney, you have the largest internal organ in your body, the liver. It sits right about here, okay? And so the right kidney has to be lower down to allow for this to exist here, okay? All right. Now, the breast. So the breast is a glandular structure. It's composed of all these mammillary glands here. And its function is to produce breast milk for babies, okay? So what's the structure of the breast? So you have these mammary glands here. Okay, these are modified sweat glands. They are going to produce a lipid-rich secretion, milk, to provide nourishment for growing babies. Okay, so you have these mammary glands. They're going to sit in and the breast milk. So when you're pregnant, these mammary glands are going to start developing. They're going to start growing, and then the milk is going to be produced and released based on like hormone released when the baby when the baby is suckling on the breast. Okay, so these mammary glands are sitting in a connective tissue stroma all around here that you can see, and that's composed of fat and fibrous tissue. You have two important fascial layers, one at the front called the superficial fascia, and then one at the back called the deep fascia. And the deep fascia sits at the back of the breast, and it's going to sit right over the pectoral muscle, which you can see right here. Okay. And that's all you need to know about the breast. It's pretty straightforward. Okay, so just know you have these mammary glands and a connective tissue stroma, and these glands are going to be producing milk for developing babies. Okay. All right, now we're going into the reproductive systems. Okay, so we're going to start with the female reproductive system. So the female reproductive system is going to be responsible for producing gametes. So in females, we call them eggs or ova. It's also going to be producing important sex hormones for females like estrogen and androgens. And it's also going to, you have the uterus, which is going to grow fetuses inside of females. Okay. So we're going to start with the internal anatomy, okay? So the first structure is the ovary, okay? And the ovary is going to be producing female gametes. It's similar to testicles or testes in males, okay? So that's what is going to produce the egg cells in females, okay? And then you have the fallopian tube. So when an ovary produces an egg cell, it's going to release that egg cell, and that egg cell is going to be collected by these finger-like projections called fimbriae at the end of the fallopian tube. The egg is going to be pulled into the fallopian tube by the fimbriae, and it's going to enter the first part, or the distal part of the fallopian tube, called the infundibulum. And then you're going to move down the fallopian tube to the ampulla, which is here, and then the isthmus, which is here. And then we go into the uterus. And the uterus is where the fertilized eggs are going to attach, and where developing fetuses are actually going to grow. Okay. And you have three layers of the uterus. The innermost layer is called the endometrium. And then you have a muscular layer in between called the myometrium. And then an outer layer called the perimetrium. Okay. And then going downward, we have the cervix, which is basically like the opening into the vagina. Okay. And then the vagina is going to be just basically a muscular tube that extends from the cervix to the outside world. Okay. And it has many roles. It's a passageway for menstrual flow during menstruation. It's used for intercourse, and this is the channel that babies are going to pass through during childbirth. And so the vagina actually has a lot of rich colonies of bacteria, which are very important for its health. They provide protection. They help maintain the pH. So the vagina is actually going to be kept at an acidic pH. It's important for cleaning, and it actually helps prevent infections as well. Okay, so that's the female internal reproductive system. Okay. And now for the external reproductive system, the external genitals, we have a couple important structures here that you should keep in mind. So you have two large folds of skin on the outside called the labia majora. And these folds of skin are going to meet superiorly at what is known as the mons pubis. Okay. And then you have two internal folds of skin called the labia minora. Okay, at the top here, you have the clitoris, and then you have the two major openings of the external genitalia, which are the urethral opening, which, if you remember, the urethral opening is where urine is going to pass through. That's the terminal, the end of the ureter. And then you have the vaginal opening, which is the opening of the vagina. Okay. All right, and now here are just some extra pictures for you. So if you want to come back, watch the recording later, I just threw in these extra pictures for you to just extra study resource for you, okay? All right, now we have another knowledge check question real quick. Where in the fallopian tube does fertilization occur? Quick throwback, if you remember from my first lecture. Wow, you guys are, I'm very impressed with this. You guys are killing this right now. And I think, you know, if you don't get this right, it's on me. I was supposed to mention this when I was going over the internal anatomy, and I don't think I did. So don't beat yourself up if you get this wrong.
You did great, Zach.
Thank you.
And so are you, students, for participating in this. All right, we got a good number of participation. We'll go ahead and close this poll in 5, 4, 3, 2, 1.
All right, yeah, so the majority of you are right. It is the ampulla. Okay, so the egg cell produced by the ovary is going to be pulled into the fallopian tube, and then when it gets to this point right here is where it's most likely going to meet a sperm cell for fertilization to occur. Okay, and this is the ampulla right here. Okay. All right, so now moving on to the male reproductive system. So similarly to the female reproductive system, you're going to have the production of sex hormones. In the case of males, it's going to be testosterone, and it's also going to produce the male gametes, which in this case are sperm cells, and yeah, and it's going to be used for sexual reproduction. Okay. All right, just want to talk briefly about the components of semen, because I think this might be a little less intuitive. So semen is a fluid that is composed of various glandular secretions. So the majority of semen is going to be fluid produced by the seminal vesicles, and I'm going to talk to you about all of these different glands in a sec. The majority of it is going to be produced from seminal vesicles. 30% is going to be from the prostate glands, and 5% is going to be from the bulbal urethral glands. And the sperm cells are just going to be residing in this fluid here. Okay. So all of this together, all of these secretions together is called semen. Okay. And then sperm are the actual reproductive cells themselves. Okay. Okay. So what is the path of sperm going to be in the male reproductive system? So you start in the testes down here. This is where the spermatogenesis is going to take place, where those sperm cells are going to mature and develop and get ready to do their job. Okay, then they're going to travel into this structure called the epididymis, which is where they're going to reside until ejaculation actually takes place. And then when ejaculation is getting ready to happen, they're going to travel up this long tube here called the vas deferens. They're going to pass behind the bladder, which is here, and in front or next to the first gland, the seminal vesicle. It's going to pass into a structure known as the ejaculatory duct. And the ejaculatory duct is going to meet up with the urethra, which is right here. Okay. And then the sperm is going to enter into the urethra and pass out through the penis. Okay. So that is the path of sperm and all the structures important to the path of sperm. Okay. Now, those glands that I talked to you about earlier. Okay, so we have 3 major glands that are very important to producing the components of semen. Okay, the first is, or we're going to talk about, is the prostate gland, which sits about right here. It's going to produce, remember, about 30% of the semen, and it's going to produce an alkaline fluid that helps protect the sperm from the acidic environment of the vagina. Remember, I told you earlier that the vagina, that the vagina is acidic, and that would be pretty harmful to sperm. But this prostate gland is going to produce a an alkaline fluid that will help protect from that environment. And then we have the seminal vesicles right here. You have two of them, but since this is a cross section, you can only see the one. And remember, that produces the majority of the semen. It's going to produce 65% of the fluid in semen. Okay, and this is just going to this fluid here is going to provide metabolic support to the sperm cells. Okay, remember they are in fact cells, so they do need some nutrients for metabolism, right? And then finally, at the end, we have the bulbal urethral glands. Again, you also have two of these guys, and they produce about 5% of the fluid in semen, and they're just going to produce a thick, clear mucus that's a part of the semen. Okay. All right. Last knowledge check question. I hope you guys get this right. I just talked about this. So which gland produces the majority of fluid found in semen? Good.
All right, we'll close this poll a little earlier because you guys have just been rock stars at participating tonight. All right, we'll close it in 5, 4, 3, 2, 1.
Yeah, great job, guys. So that is going to be the seminal vesicle. So that's going to produce about 65% of the fluid found in the semen. Okay, so great job. All right, and that is all that I have. So now we can get into some questions. Let me just pull up the chat here.
And again, to our participants, feel free to type in the chat or feel free to raise your hand and ask the question verbally. But kind of anything you want to know about reproductive anatomy for Zach.
Okay, let me back to the uterus slide here.
And then just to read the question out loud for our recording, we got a question from one of our participants asking, what is the purpose of having 3 layers in the uterus?
So yeah, so I mean, obviously nothing with the human body is like intentionally designed. It just kind of evolved that way. So this inner layer is kind of like a regenerative layer. So we call it the endometrium. That is where the egg is going to, the fertilized egg is going to attach. And during menstruation, females go through cycles of growth of that layer. And the growth phase is basically preparing for fertilized egg attachment. Okay, so you need to get all the nutrients that an egg would need in order to, be healthy and to grow and to survive. And then you have a muscular layer that basically evolved to allow for contraction of the uterus. Okay, and the uterus needs to contract for various reasons. So like during menstruation, if there's no fertilization of the egg, the uterus is going to contract and expel all of that tissue that it grew in preparation for attachment of the fertilized egg. And then you have an outer layer, which is, I think, just like a connective tissue layer that attaches the uterus to like surrounding structures and keeps it nice and in place. Okay, so anatomists decided to separate those 3 divisions based on the function that they served for the uterus.
Before you move on, Zach, there's actually a point Dr. Peterson, I think, brought up in last year's session. Can you just identify for students where implantation occurs in those layers?
Yeah, like which layer it occurs at? Or like where?
Maybe
Like
a little of both.
anterior.
Yeah, a little of both, Zach.
I think it typically occurs on the anterior wall of the uterus, right?
So typically, posterior and more
Oh,
superior.
okay.
The main point is that we don't want it to be really inferiorly localized, because again, if in fact this is an implantation event and we start to have a growing embryo and then a growing fetus, it's growing within the wall of the endometrium. And I think a lot of females who have actually given birth don't understand that. Many females think that the fetus is just developing within that cavity, when in fact it is not. It is developing within the
Yeah.
innermost layer of the uterus. And so
Mhm.
if implantation occurred low, over the opening of the cervix there, that could be an issue once a parturition or childbirth was imminent. So,
Yeah.
yeah, I think you have another question in the chat,
Yeah.
but maybe it might be a little bit more appropriate for what Maddie is going.
Yeah. Yeah. So we're Maddie is going to go over the histology of the urinary system. So she'll cover what kind of epithelium is there. So that if you want, you can ask that when she presents. Or I mean, I can answer it to whatever works. Any other questions?
Okay, it doesn't look like there's any, Zach.
All right.
So we'll let you off the hook.
Okay,
So once
great.
again, excellent presentation, and thank you again for all the time and effort that you've spent here, especially for this year's Anato-Bee. So job well done.
Thank you.
With that, we will move on to our next presenter, who will be talking about the histology of the urinary and reproductive systems. So I want to introduce Maddie Sarnac. Maddie is from Fort Myers, Florida, and graduated from Florida State University in 2020 with a Bachelor of Science in Interdisciplinary Medical Sciences. She is currently a second-year student at Edward Via College of Osteopathic Medicine in Auburn. In her free time, she enjoys trying new recipes, watching movies with her cat, and we need to find out what your cat's name is, and then spending time with friends. Uh, Maddie's favorite part of anatomy is neuroanatomy. So, Maddie, it's all yours.
Thank you. I'm just going to go ahead and share my screen. Oh, okay. All right. Okay. Hi, everybody. Today we're going to be talking about histology of the urinary and the reproductive systems. I just wanted to give a shout out to Mackenzie Blackstock. She presented this lecture last year, so just a huge shout out for all the work she put into the PowerPoint. These are going to be your lecture objectives. We're going to talk about the microstructure of the ureter, of the urinary bladder, and the urethra, and how that correlates with their location and their function. We're also going to talk about the gross histological organization of the ovary, and we're going to compare the histology and the function of the 3 layers of the uterine wall. Lastly, we're just going to talk about the microscopic organization of the testes and how that correlates with their function. Like Zach said, when we refer to male and female within this lecture, we're referring to biological sex and not gender. So when we refer to male, we're talking about testosterone-driven anatomy, and when we refer to female, we're talking about estrogen-driven anatomy. So just to get started, I know you guys just saw this, but to review for the anatomy of the urinary system, you have 2 kidneys, you have your 2 ureters, and then you have the bladder and the urethra. And so when we talk about the histology of the ureter, we're focusing on its 3 layers. The first layer we want to talk about is the outer adventitia. And this is composed of fibrous connective tissue, and it houses all the blood vessels that help nourish the ureteric cells. And it's this layer right here. It is also going to help anchor the ureter to the posterior abdominal wall. Moving towards the lumen, the next layer is the middle muscularis layer. And this layer has your smooth muscle. And that smooth muscle is going to help the ureter propel the urine from the kidney all the way down to the bladder. And it's all of this muscle right here. The last layer we want to look at is the inner mucosa. And the inner mucosa surrounds the lumen. Some people kind of describe it as having a star shape, the lumen. And the inner mucosa is going to be lined by something called transitional epithelium. And we'll see what transitional epithelium looks like up close in a couple of slides. But the inner mucosa is going to function to help secrete a mucus. And that mucus also allows for expansion and contraction so that the urine can pass through the ureter and get down to the bladder. So when you're thinking about the ureter, you just want to think about the outer adventitia, the middle muscularis, and then the inner mucosa. When we look at the bladder histology, it's very similar to the ureter, but it has a couple of exceptions. One being that the superior surface of the bladder is covered by a serosa, and that serosa is just a single cell layer of simple squamous epithelium. The next is that the outer walls of the bladder are covered by an adventitia, but the main histological difference and a very key histological point of the bladder is that it's composed of 3 layers of smooth muscle. It has an inner and an outer longitudinal layer, but then it also has that middle circular layer. So when you think about the bladder and its muscularis layer, it's quite thicker. The mucosal layer of the bladder is the same to that of the ureter in that it's made up of transitional epithelium. So what is transitional epithelium? First, I just want to review stratified squamous epithelium to kind of give you something to contrast transitional epithelium with. But if you look at stratified squamous epithelium, you can see that it's got this flattened outermost layer, and then it has many layers below that. And we tend to find stratified squamous epithelium in areas like our skin. And when we think about what we want our skin to do, we want it to protect us from from any harm or from the environment, so that our deeper tissues are safe. So let's say you're playing with your cat, they scratch you. We like the fact that our skin has a lot of layers because it protects the deeper tissues from any harm that could come from the environment. When we look at transitional epithelium, we can also need to take into account where it's located and what the function of that organ is, and it can kind of help us understand it. So transitional epithelium can look as stretched or distended, which is this middle image, or it can have this unstretched, relaxed appearance. However, transitional epithelium still has that flattened outermost layer, but it's got fewer layers inside, especially with the stretched transitional epithelium. And the reason for this is as the bladder collects urine, we want it to have the ability to grow and distend. We don't want the bladder to not have the ability to grow because that could cause problems with us being able to having control over using the restroom. So when we think about the tissue that is within the bladder, we want it to have that ability to grow and distend. So we want it to have fewer layers. Now, once you've used the restroom and your bladder is not distended or it's not stretched, it does look like it has many layers, but it's still not going to have the same amount as a stratified squamous. A really key difference when you're trying to compare, am I looking at stratified squamous versus if I'm looking at unstretched, is you can look for something called a balloon cell. You can see them here on this image to the right. The balloon cells look a bit circular. They kind of look like full balloons, maybe with a little eye decoration in the middle of them. But that can kind of help you tell the difference between the many layers of the stratified squamous and some of the layers of the unstretched transitional epithelium. Now with those 2 questions, we're going to go ahead and put up a poll. So the first question is, what is the order of the layers of the ureter? And we're working from the outermost layer all the way into the innermost. Okay. Yeah, it looks like you guys are getting it.
All right, we'll go ahead and
So,
pull. Sorry, Maddie, we'll close this poll in
Oh, great.
5, 4, 3, 2, 1. You guys have been doing such a great job with participating tonight.
yeah, you guys are on fire tonight. So a lot of you answered A, which is the correct answer. So when we think about the order of the layers of the ureter, the outermost portion is the adventitia, middle muscularis, and then the mucosa is going to line the lumen. So good job, everybody. And then the second question is just, what is the urinary bladder's muscularis layer composed of?
I don't know if anyone here likes Jeopardy, but I keep just imagining that song in my head during that quiet time. We need some music next year.
Yes,
And then
next time we
polling.
get the transitions.
Yeah.
Yeah.
All right. We'll close this poll in 5, 4, 3, 2, 1.
Good job again, everybody. So the correct answer this time was B. So remember that within the bladder, it's the muscularis layer. There's three layers of muscle, and the inner and the outer are longitudinal muscle, and it's that middle layer, that circular muscle. But you guys are doing awesome so far, so we'll keep going. So the next part of the histology of the urinary system we're going to talk about is the urethra. So as Zach talked to you guys about, the female urethra is a lot shorter. It's a lot smaller than that of the male, and it is composed of a mucosa and a muscularis. So when we look at the mucosa, the type of epithelium, it varies greatly, but it does have longitudinal folds. And then in this bottom right image, you can see the urethral glands that can be found in the female urethra. The muscularis layer of the female urethra is composed of a longitudinal and a circular layer of smooth muscle. But unlike how we saw in the ureter and the bladder, which both had a clear adventitia, we don't necessarily see that within the female urethra. When we talk about the male urethra, we have to think about the anatomy that occurs. So we know, because Zach just taught it to you guys so well, we know that there's different portions of the urethra. And when we think about the histology, we have to first ask ourselves, where are we? And the reason for that is because the epithelium changes based off the location. So that very first part of the urethra coming right out of the bladder into the prostate, that's your prostatic urethra, and it's composed of transitional epithelium. So a lot like what we saw within the bladder and then within the ureter. It is resting on a lamina propria, and it does have as well urethral glands. When we talk about the membranous urethra, so we've left the prostate, and we keep going down, the membranous urethra is going to be made of pseudostratified columnar epithelium, so it changes. Also, a fun fact about the urethra is that within the prostatic urethra and the spongy urethra, there's no muscle present. However, in the membranous urethra for the male, skeletal muscle is present, and it's present at that level of the external urethral sphincter. And in the membranous urethra, we're going to find bulbal urethral glands. Finally, in the spongy urethra, and that is the portion of the urethra surrounded by that corpus spongiosum, or your erectile body, the spongy urethra is composed mainly of pseudostratified columnar epithelium, a lot like what we see within the membranous urethra. However, as we get to that terminal portion of the urethra, close to the end, we have stratified squamous epithelium, so it changes for a third time. So when you're thinking about the male urethra, you just want to kind of first ask yourself or look for, where exactly are we located? Because the epithelium can change, and it does change. So with this next review, I just want you guys to put in the chat, which portion of the ureter do you think that this arrow is pointing to? Let me see if I can get the chat back up. Or if we can't remember that, or we're having some trouble remembering that far, we can kind of go over it together. Awesome. Yep. Perfect. So yes, it is the outer adventitia. You guys are doing great tonight. I don't even think you need me. What layer is this arrow pointing to? Perfect. Awesome. Okay, and finally, how about this layer? Yep. Good job, everybody. Okay. Can we identify this structure right here? And then bonus question, if you remember what type of epithelium we can find there. I'm seeing a lot of prostatics, and that would be correct. So that's your prostatic urethra. And then the epithelium that is found there is the same that's found within your bladder and your ureters. So it's your transitional epithelium found within the prostatic urethra. Good job, guys. All right, we're going to keep moving along. So just a quick review about the female reproductive anatomy. So these are your internal female structures we're concerned about, the first being the ovary, and then we have the fallopian tube. We've got the uterus right here, and then we've got the upper portion of the vagina, and that's all the internal female structures. So when we look at the ovary, there's 4 layers that we're concerned about. The first one is colored here in blue, and this is your germinal epithelium. And what we think about it as is just the envelope of the ovary. It encompasses the entire structure, and it's made of simple squamous to cuboidal epithelium. The next layer is here in the green, and this is your capsule or your tunica albuginea, and it is going to be the layer that really protects all the deeper structures within the ovary. And we'll see that word again in a couple of slides, but when you see tunica albuginea, you want to think protective layer. It's going to be composed of fibrous and collagenous layers as well. As we move closer to the middle of the ovary, we've got the cortex. And the cortex is going to be where your ovarian follicles live, and that's going to contain your eggs. It's also got some connective tissue elements here, so this is all cortex. And then finally, we've got the medulla, and the medulla is going to be an area that's highly vascularized, so it's got a lot of blood supply, and that blood supply brings important nutrients to all of these structures here. So when we take a closer view at the cortex, we can see all the follicles, which are in various stages of development. So out here on the outskirts, you can see primary follicles coming into secondary, all the way to tertiary and mature. Now on your anatomy exams, you're not going to be asked to identify where in follicular development a certain follicle is. I just thought it was a cool image to show you all how within a single ovary, you can see each follicle in different stages of development. And this all occurs, all your follicles live in the cortex of your ovary. Now, when we talk about the uterus, which is this structure right here in the yellow box, it's got a couple different layers to its wall. The first one being the perimetrium, and that's, if we think about the outside of the uterus working our way in, the very first layer closest to the outside of the uterus, that's your perimetrium. The posterior surface of the perimetrium, it has a serous layer, and that serous layer actually helps protect the uterus from any friction that it can occur while within the abdomen. It's part of a larger structure called the peritoneum. That's a fold that helps protect all of the structures in your abdomen, and it's made up of mesothelium and a loose connective tissue. And then the anterior surface here of the uterus is your adventitia. As we move closer to the inside of the uterus, the middle portion is the myometrium. And you can see in the bottom image how the myometrium actually takes up a large portion of the uterus. It is of the uterine wall, excuse me. It is quite a bit. This is a really important layer composed of 3 layers of smooth muscle, and it functions to help expand in pregnancy. And then this is where your contractions are occurring and during childbirth to actually expel the baby from the uterus. So this is a very important layer within the uterus. And then finally, we have the endometrium. Now the endometrium is the innermost layer of the uterus. It is going to be the layer in which if an egg is fertilized, this is actually where it implants. It's made up of simple columnar epithelium with a mucous membrane. And there's 2 main layers of the uterus that are really important, one being the functional layer. Now throughout a menstrual cycle, the thickness of this endometrium will change and it'll actually get quite thick. And then if fertilization does not occur that cycle, it will shed. And that's what menstruation is. It's the shedding of this endometrium. It's your functional layer that's growing in thickness and then shedding during menstruation. The basal layer, that remains relatively constant. And all it's doing is it's serving as a source to the functional layer so that it can constantly regenerate and have something to build up on and then shed when fertilization doesn't occur. So functional layer, what is shedding, basal layer, your source for regeneration. This top image here just shows you how the histology of the endometrium changes as you progress throughout the uterine cycle. Again, you're not going to be responsible for identifying this is this phase and this is this phase on your exam. I just thought it was cool for you to see how much can change based off where someone is within their cycle. With that, we're going to do a couple of polling questions. So the first question is, in what layer of the ovary are the ovarian follicles located?
All right, we'll go ahead and close this poll in 5, 4, 3, 2, 1.
Okay, awesome job, guys. So a lot of you answered C, which is the correct answer. It's the cortex, was where the follicles are. Remember that the capsule is going to be your protective layer for all of your deeper structures. So the capsule is close to the cortex, but it's not where the follicles live. It's actually the area that helps protect the follicles. So the next question is going to be, in which layer of the uterus expands during pregnancy and contracts for childbirth?
You guys are really getting your prefixes down. All right, we'll close this poll in 5, 4, 3, 2, 1.
Awesome job, everybody. So the correct answer here was also C. Remember that it's the endometrium where the egg will implant if fertilization occurs, but it's actually the myometrium that will contract during childbirth and during labor. But awesome. We're going to move on to the male reproductive system, and we're going to talk a little bit about the production of sperm. Remember that sperm is going to be produced within the testes, and we'll get into what the seminiferous tubules look like at a histological point of view. But sperm will be produced within the testes, and then it will move through the epididymis up to the vas deferens, ductus deferens, ejaculatory duct, and then eventually we'll get to the urethra. So when we talk about the histology of the testes, the first layer we're going to talk about is the tunica albuginea. And if you're saying, I just saw that word, you'd be absolutely correct. It's also what we call the capsule within the ovary, and it has a very similar function. It is going to help protect all of your underlying structures. And it's also made up of fibroblasts and collagen fibers. So again, when you see that word, start to think protective layer. We also have Leydig cells. So in this image on the right, you can see the Leydig cell. And the Leydig cell's really, its main job is just to produce testosterone. And in its production of testosterone, it helps sperm get produced. The seminiferous tubules can be seen on both images. The one on the right is a bit of a closer look at a seminiferous tubule, but it helps produce sperm through a process called spermatogenesis. This is actually where sperm gets produced. So if we looked at it at a more zoomed in version, the seminiferous tubule is made up of epithelium and it contains Sertoli cells, but these cells will envelope and support the sperm at various stages of their development. So we can see on the image in the right, a lot like we saw within the cortex of the ovary, sperm at various stages of development, whether it be a spermazoa, which is your mature sperm, or a spermatocyte or a spermatid. We can see all of its stages of development, and you can kind of notice how we go from the more immature or the more early development is on the outskirts, and as we move in, we've got more mature or sperm in later stages of development. The Sertoli cells are here on the outskirts. Your Sertoli cells are also referred to as nurse cells. The reason for that is they're just providing support. Their job is to help protect the sperm from anything in the body that might destroy it or might harm it, and help it so that it can develop into a spermazoa to then be used to fertilize an egg. So with that, we've got one more. I'm sorry, my cat's trying to make her debut on the Zoom. We've got one more review. So I just want you guys to put in the chat, what do you think the structure, which the arrow A is pointing to, which of the following, 1, 2, or 3, do you think is the function of that structure? And if I need to rephrase that, I can. I think I kind of said that in an odd way. So that is correct. A is 3. We're looking at a seminiferous tubule, and that is the site of sperm production. So good job, everybody. What number, 1 or 2, do we think B is? Yep. Okay. Oh, I gave one away. See if I can get that out of here. But B is 2. The reason we're looking at is we're not necessarily looking at the cortex. We're looking at the level just above it, which is the capsule, which is the tunica albuginea, and that is going to be the protective layer of the ovary. And so what would that make C? Yep, C is 1. It's the mucosal layer, which helps accommodate for changes in volume. And with that, those are my references. And that is my cat. Her name is Phoebe. She is very excited for Christmas. And does anyone have any questions for me? I can get the chat up.
One of the questions we had earlier that I can return back to was what would happen if the urinary bladder didn't have transitional epithelium? How would that impact its ability to stretch?
Let's see if I can find that slide. For example, if we were looking, if the urinary bladder for some reason had maybe stratified squamous epithelium and it had those many layers in its inability to stretch, the bladder wouldn't have the ability to distend and hold urine, so we wouldn't be able to, like, you're on a long car trip and you need to hold your urine, there wouldn't be anywhere for it to go, so you would have to expel it. So our epithelium there helps us, the bladder distend so that it can hold the filtrate until we're able to actually expel the urine.
Maddie, it's Dr. Peterson. I'll just jump in and then maybe Dr. Mapes will want to add to this. But it's really important that the lining of the bladder or these different tubes that are carrying urine, that it has the same capacity to kind of match what's happening in the muscularis layer. So smooth muscles are much better at being able to relax or contract. And epithelial typically does not do that. And so this is really unique and a special feature. Typically, what we want epithelial cells to do is just be held really tightly together in sheets. And then sometimes those sheets are comprised of multiple layers. So this is really unique and special. And that's why we make such a big deal out of it in the urinary system. But you did a great job of explaining that.
I guess since you asked Dr. Peterson, I guess the only other thing that, we kind of didn't talk about, we talked about its ability to stretch, but it does also offer a layer of protection, right? All those layers, and there's some special proteins up there that help, because urine, pH is a little bit different. There's maybe different osmolarity, and so that layer, that special transitional epithelium, not only allows for that ability for the epithelium to move with the smooth muscle, but also adds a kind of extra layer of protection there from that urine.
We have a question in the chat. What is the anterior layer of the perimetrium in the uterus?
I'm not entirely sure, actually. I know that in one of our slides, we talked about how, let's see if I can get back to it. Within the uterus, the perimetrium, their posterior surface was the serous layer, which helps protect from friction, and the anterior surface is just the adventitia, or the outermost portion. I don't know if that was the question. I don't know if there's different layers. There might be different layers within the perimetrium that we didn't talk about, but I just know the difference between the two surfaces.
Maddie, maybe just to add on to that, just many of the reproductive organs in the female are part of a ligament, which is a really a poor label to put on it, called the broad ligament. And it's almost like a sleeve that completely is surrounding these reproductive structures, but it's really more like a mesentery. And maybe those of you watching the video or those of you here with us in person will remember that the real function of a mesentery is to bring the blood supply and the lymphatic supply and the nerve supply to these organs from the abdominal wall. So that's another sort of additional layer above and beyond what you've helped describe to everyone tonight.
Okay, any more questions for Maddie? Okay. If there are none, we can move on. Thank you, Maddie. Excellent presentation. So our third and final presenter will be talking about the embryology of the urinary and reproductive systems, placenta and fetal membranes. Dominic Finan is from Malvern, Pennsylvania, and graduated from the University of Scranton in 2023 with a BS in neuroscience and philosophy with a minor in biochemistry. He is currently a second-year medical student at the Sidney Kimmel Medical College at Thomas Jefferson University. Dominic enjoys running, trying new restaurants, and painting. His favorite part of anatomy is cardiopulmonary anatomy. So with that, Dominic, the floor is yours.
Thank you so much. All right, everybody. Hello. Today, I'm going to be talking to you about the embryology of the urinary and reproductive systems. So can everybody see my screen properly? I just want to make sure everybody can see it. Good? Wonderful. So these are our learning objectives. So when you guys are studying this presentation, I want to make sure that you stay within these learning objectives, just because I know embryology can be very intense. Even as a medical student, I still get super confused about embryology. So I wanted to make this presentation pretty basic and pretty close to what these LOs are testing. So when you're studying, please just refer back to these. So what we're going to be learning about today is we're going to be describing the development of the pronephros, the mesonephros, and the metanephros. We're going to be discussing the formation of gonadal ridges and genital ducts. And finally, we're going to be talking and discussing the development of genital ducts in the presence or absence of testosterone. So beginning with the urogenital system. So the urogenital system is derived from the intermediate mesoderm. So after gastrulation into the trilaminar disc, the mesoderm actually splits into three separate components: the paraaxial mesoderm, the intermediate mesoderm, and the lateral plate mesoderm. So this presentation is going to be focusing on this intermediate mesoderm because that is the layer from which the urogenital system derives. So the intermediate mesoderm condenses and becomes the genital or gonadal ridge and also the nephrogenic cord. And I'll talk to you more about these specific terms a little bit later, but just remember from this slide that the urogenital system emerges from the intermediate mesoderm. So this is just a basic outline of weeks 4 through 12 of development. So when I use the phrase nephro, what I'm referring to is the microscopic functional unit of kidney filtration. And there are three different sets of primitive kidneys that develop. We begin with the pronephros, the mesonephros, and then we end with the metanephros. And this metanephros is what persists as the adult kidney. The metanephric duct actually forms as an outgrowth of this mesonephros at its most distal end. And I will come back to this slide at the end after I discuss the pronephros, mesonephros, and metanephros. So again, let this be an outline that helps you come back to when you are studying. So beginning with the pronephros. So around week 4, the cervical region of the nephrogenic cord, which is from the intermediate mesoderm, develops into the pronephros. So this image is what I'm going to be using the entire time. So this is a 5-week embryo, and what we are referring to right now is the pronephros. And when I use the phrase cervical, what I mean is the portion that is closest to kind of the head of this embryo. So in humans, the pronephros actually never forms functional nephrons. This pronephros is more or less a scaffolding from which more important structures derive from. So let this be basically a precursor to the mesonephros and the metanephros, which are the more important structures, and those actually persist into adulthood. So by the end of week 4, the pronephric tissue actually degenerates completely, and the mesonephros begins to develop from that. So moving from the pronephros, now we have the mesonephros. So we're moving down from the cervical region and we're moving more towards the thoracic or lumbar regions. So by the end of week 4, the mesonephros begins developing from this degenerating pronephros. And as I said, it develops in the thoracic or lumbar regions of this nephric duct and induces a new set of tubules or glomeruli and connects to the cloaca. And when I use the phrase cloaca, I refer to that which allows waste to actually be removed from the embryo. So it develops into something that allows liquid and solid to be removed from the embryo. And these actually form functional nephrons, and they're able to produce urine. And they're actually the main producer of urine up until around week 10. And as we will see a little bit later, around week 9 is when the metanephros is able to develop fully. And the metanephros is what persists as the adult kidney. So while the metanephros is developing and growing, the mesonephros is what filters the blood and filters the blood from toxins and is able to actually make urine. So during this time, the mesonephros is, for all intents and purposes, the urinary system in the growing embryo. Now, in my next slide, I'm going to be zooming in to this portion of the embryo in the most kind of distal portion. This is the metanephros. This is the most important portion because this is what persists as the adult kidney and as the adult urinary system. So now moving on to the final portion, we are at the metanephros, and this develops from the most caudal intermediate mesoderm. And as I said before, this is what becomes the permanent kidneys, and it begins developing around week 5, but it is not functional until around week 9, because if we remember from my last slide, between weeks 5 and 9, the mesonephros is what is able to form urine and able to remove toxins from the blood. So while this metanephros is developing, the mesonephros is basically doing what the metanephros will do in adulthood in this embryo. And this nephric duct actually grows into the metanephrogenic blastema and forms this ureteric bud. And this ureteric bud becomes the collecting system, and the metanephric blastema forms the permanent nephrons, which is where urine is produced. So I know these are a lot of words, so I'm going to try to break it down into simple terms. So this metanephric blastema is what the blood flows into. And when the blood flows into this metanephric blastema, the tissue is able to remove the toxins. And when the toxins are removed and the waste product is removed from the blood, it is transferred into this ureteric bud and into this ductal system. So these ductal systems are more or less a highway to allow the toxins to be removed, but this metanephric blastema is what's actually doing the work and what is actually removing the toxins. And once it does so, it transfers the liquid or the urine into this ureteric bud, and then it flows from the ureteric bud through the ductal system, and then eventually out of the cloaca and out of the body. So now we have a quick quiz. So from which embryonic layer do the kidneys form? Okay, it looks like we're waiting on a little bit more. Okay.
Alrighty, I will close this poll in 5, 4, 3, 2, 1.
Awesome. Good. Okay, so yes, it comes from the intermediate mesoderm. So if we remember, the mesoderm develops into 3 separate portions: the paraxial, the intermediate mesoderm, and then the lateral plate mesoderm. But the kidneys and the urogenital system derive from this intermediate mesoderm. So it looks like about half of us got that. Awesome. All right, next. Which embryological structure persists as the adult kidney? So essentially, which of these remains as the adult kidney?
All right, get your final answers in. We will close this poll in 5, 4, 3, 2, 1.
Awesome. Yes, the metanephros. So if you remember, the metanephros is the last structure to form. It forms in this most kind of caudal region, and this is what persists as the adult kidney. Okay, so let this be a quick summary of what happens between weeks 4 through 12. I know this is a pretty busy slide, but it's more or less just for kind of studying purposes. As I said before, the phrase nephro refers to the microscopic functional unit of kidney filtration. There are 3 separate forms of primitive kidneys that develop. It begins with the pronephros, and this is not necessarily important for human adult development because the pronephros are just a apparatus or a scaffolding from which the mesonephros and the metanephros derive from. The mesonephros is only important in testosterone-driven anatomy because, as we will see in the second portion of my presentation, this persists as the tubular system that connects the testes to gamete outflow. And then finally, the metanephros gives rise to the adult urogenital system. And these structures will essentially connect with the developing bladder and persist as the urinary system. The distal end of the gut tube is what we refer to as the cloaca, and it is separated into two different reservoirs, one for liquid waste and one for solid waste. So in terms of this presentation, we were referring to kind of this liquid waste reservoir and what the ducts kind of drain into, which will essentially be the bladder. This is that portion of the cloaca. And then, as I said, the metanephric duct forms as an outgrowth of the mesonephric duct at its distal end. And this metanephric duct will induce caudal intermediate mesoderm to form the metanephric blastema. And this metanephric blastema is the true filtration unit. This is the nephron. This is where the blood will flow into. This is where the blood will be filtered. And once it is filtered into urine, the urine will flow into this ureteric bud. And then in this ureteric blood, the urine will be drained into the bladder and then essentially removed from the body. So now moving on to kind of the genital system. So we're going to start with gonadal ridges. So the development of gonads are actually bipotential initially. And when I use the phrase bipotential, I mean it can develop either into testes or into ovaries. At week 5, there is a thickening of tissue ventral to the mesonephric ridge, which forms the genital or gonadal ridge. And we can use those phrases interchangeably. Germ cells, which are another phrase for reproductive cells, actually migrate to these gonadal ridges at around week 7. And then after this migration, the formation of genital ducts are dependent on the presence or absence of testosterone. So essentially, both sexes start with two ductal systems. So much like the gonads, the ducts that connect these gonads to the outside world, for lack of a better term, are bipotential. So they persist and they exist kind of together, but only one will persist in the end. And the two different ducts that can form are the mesonephric duct or the paramesonephric duct. And this mesonephric duct, another name for it is the Wolffian duct, and I'm going to be using that phrase Wolffian duct for the rest of this presentation, but just know that Wolffian is what derives from the mesonephric duct. And this is what persists in XY embryos or in testosterone-driven anatomy. And this Wolffian duct is what becomes the seminal vesicles, the epididymis, the ejaculatory duct, and the ductus deferens. Conversely, the Müllerian duct, which is what persists in XX embryos in estrogen-driven anatomy, this is what becomes the fallopian tubes, the uterus, and the proximal vagina. But as I said, although both sexes start with both of these ductal systems present, only one ductal system persists, and which ductal persists is essentially contingent on which hormones are present, and the hormones are made either by the testes or the ovaries. So depending on what hormones are made, it decides whether the Wolffian duct persists or the Müllerian duct persists. So this is a flow chart. As I said, the gonads and the ducts begin as bipotential. So they can either become the ovaries or the testes. In the presence of testosterone or in the presence of testes, the Müllerian ducts will degenerate and the Wolffian ducts will persist. And these Wolffian ducts will become the seminal vesicles, the epididymis, the ejaculatory duct, and the ductus deferens. However, in estrogen-driven anatomy or in the absence of testosterone, these Wolffian ducts will actually degrade and the Müllerian ducts will persist. And these Müllerian ducts become the fallopian tubes, the uterus, and the proximal vagina. So this is kind of an overflow or an overview or flow chart of the bipotential system. So beginning with the Wolffian duct, so there is an SRY gene on the Y chromosome that produces testes determining factor. And this is what stimulates testes development. So without this SRY gene and without the Y chromosome, testes cannot develop. And the testes have two important cells that ensure that the Wolffian duct develops and the Müllerian duct degenerates. And these two cells are the Leydig cells and the Sertoli cells. So the Leydig cells create something called androgens, which for this presentation, we're going to be focusing on testosterone as the most important androgen, which is a hormone. Testosterone stimulates the development of the Wolffian duct, whereas Sertoli cells make a different hormone, anti-Müllerian hormone. And based on its name, we know that the anti-Müllerian hormone will suppress the development of Müllerian ducts. So the testes make two very important hormones, testosterone and anti-Müllerian hormone, and together they ensure that the Wolffian ducts persist and the Müllerian ducts degenerate. Now switching gears to Mullerian duct formation. Without the Y chromosome, we do not have the testes determining factor and we do not have testes. Therefore, both testosterone and anti-Mullerian hormone are not produced. So with two X chromosomes, we have genes that encode for ovaries. And without anti-Mullerian hormone and without testosterone, the Mullerian ducts persist and are stimulated by estrogens from the ovaries. And without testosterone, the Wolffian ducts will degenerate. So I know this can seem super confusing, but I'm going to try to kind of bring it all together and break it down in this flow chart. So we start with a primitive gonad, and this gonad is bipotential. It can either develop into the ovaries or the testes. If the embryo is XX, no testes will be developed and no anti-Mullerian hormone or testosterone will be produced. If there's no anti-Mullerian hormone, the Mullerian ducts will persist because there's no hormone telling it to stop developing. Similarly, without testosterone, the Wolffian ducts cannot persist, and the Wolffian ducts will degenerate. And in the absence of anti-Mullerian hormone, the Mullerian ducts will persist, and this embryo will develop with fallopian tubes, a uterus, and an upper vagina. Conversely, if the embryo is genetically XY, they have the SRY gene that encodes for testes determining factor, and testes are made. And these testes have two very important cells, Sertoli cells and Leydig cells. The Sertoli cells make anti-Mullerian hormone. This hormone tells the body, do not make the Mullerian ducts persist, do not let them develop. So with this hormone, the Mullerian ducts will regress. Similarly, in the presence of testosterone, which is made from these Leydig cells, the testosterone says, keep the Wolffian ducts developing, and these Wolffian ducts will develop into the epididymis, the vas deferens, and the seminal vesicles. So again, in XX individuals, we have no anti-Mullerian hormone and no testosterone, which allows the Mullerian ducts to persist. But in XY individuals, in the presence of testosterone and in the presence of anti-Mullerian hormone, the Mullerian ducts regress and the Wolffian ducts persist. So moving on to our final knowledge check. I'm not sure if the Zoom will allow you to do multi-select. So if not, just click one that you think fits. So which of the following structures develop if there is anti-Mullerian hormone and testosterone present in the genital ductal system? So in an individual with XY chromosomes.
Get your final answers in for our final polling question of the night. 5, 4, 3, 2, 1.
Awesome. Yes. So if there is anti-Mullerian hormone and testosterone present, we know that the Wolffian ducts will persist and the Mullerian ducts will degenerate, and the Wolffian ducts develop into the seminal vesicles, the epididymis, the ductus deferens, and the ejaculatory duct, but that's not in the question. So great job, everybody. And I believe that is everything. These are my references. If anybody has any questions, I'd be more than happy to answer it. I know embryology is a very complicated concept, but I hope this presentation made it a little bit more simple, but you guys are all doing incredible learning this so young. So if you have any questions, I'd be more than happy to answer any of them.
And I know asking questions about reproductive anatomy, histology, or embryology can feel a little uncomfortable, and you might feel silly asking questions like you think you should know that, but I promise you we all start somewhere. I didn't know the answer to anything about how the human body worked when I was in high school, so I promise that all of us understand. And again, now is your chance that you can ask some content experts. If you've ever just wondered how something works, feel free to drop it in the chat. And thank you, Dominic, for I think you did just such an excellent job of also summarizing everything and doing things in a nicely repetitive way so that we could really hammer it home, but not in a really good way. I think it was I think that was so easy to follow, and I appreciate you doing that with embryology. That's a hard thing to do.
Thank you. I appreciate it.
It looks like we got one question here. Is the point when the other duct persists, other regressing, when they are able to identify the biological sex of the embryo? And what week is it?
That's a good question. I know in terms of identifying the biological sex of the embryo, that usually relies on multiple different modalities, whether that be an ultrasound or whether that be genetic testing of the embryo itself. So it's very different. It depends on what test they use. And I know when a test, say like an ultrasound, looks at external genitalia, that's different than these ductal systems, because these ductal systems are primarily the internal genitalia of an embryo. So these ducts wouldn't necessarily allow a person to say this is a biological XX or biological XY individual, because the ultrasound only looks at external genitalia. And I'm not sure exactly what week you're able to see the external genitalia, but if you're doing a different test, like a genetic test that allows you to see the genetic makeup of an embryo, that will allow you to say whether or not that embryo is XX or XY. And that can be done, I think, between 10 or 13 weeks of development if you're doing like a chorionic villi sampling.
We'll check another minute to see if any questions come through.
Maybe while everybody is thinking about any last or final questions that you want to ask to any of the presenters, I think it's really important as you become reproductively mature adults that you have this fundamental knowledge, and that would be that from start to finish, not very well explained, even in college biology courses, is that a female, the reproductive gamete, the egg cell, takes almost a full year to develop. So there's lots of things that can happen to a female in that year leading up to the ovulation of a mature ovum. So that's really a consideration for anyone that's considering it may be your, individuals in your family, etc., but that you really have to think about good, good hygiene, good health habits, good nutrition, almost a year in advance of when you are hoping to become pregnant. And for men, that whole process of spermatogenesis from start to finish takes about 3 months. And that same holds true for men that are considering starting a family. We rarely talk about that, but you should be sort of edified with that information to make those appropriate life choices. So just wanted to put that out there.
I feel like if anything, Dr. Peterson, I just want like a podcast where you just like put out all these really, great concise facts about, I feel like just everything from head to toe. I always appreciate the things that I learned when you contribute those things in there too, and even from our student presenters that sometimes find fun facts that I had no idea.
Let us not forget Dr. Frank and Dr. Mapes and what they've added to this session. So thank you again to all the presenters, for everyone that was tuned in with us. And we're just hoping that your studying, your preparation efforts are going well. And we're here to support you as you move forward towards the local competition. And again, that will happen on January 25th, which is a Saturday. And we'll send out more information to your local host institutions as we get closer to that date. But we want you to, of course, enjoy the holidays, but maybe study a little bit as well. All right.
All right, thanks everyone again. One more announcement that I think was missing from the start part of this as we close this up is that All of our participants last year received a certificate of completion for attending and participating in the local competition. So all of you that participate and show up and compete on that January 25th day will receive that certificate as well. And that's something that has a trackable code attached to it. So you can include that with your undergraduate college applications, and they can verify that you in fact were a participant, and refer back to, our website then to see all the things that you did triumph through. So, and then one more question, since we're here still, is how does the embryo, how are they able to excrete their waste if the kidney and all other organs aren't developed yet? It's a good question.
It's a very good question. So between weeks 5 and 9, while the metanephros is developing, or the permanent adult kidneys, the mesonephros is able to make urine, and that urine is able to be excreted through the cloaca, and that cloaca is basically the exit portion of the embryo where both solid and liquid waste are able to be excreted. And then once the metanephros past week 10 persists and the adult kidney is being formed, the metanephros takes over from the mesonephros and then does the same thing and is able to excrete it through the cloaca. So that is a very good question.
Dominic, are you okay if I just add a few more thoughts to that?
Of course.
So just remember, everyone, that really the nutrition is coming through the maternal blood supply, and then is early on is crossing the placenta just sheerly by diffusion. So this is all at the molecular level. And then once we have the primitive heart and we have primitive blood vessels that the embryo itself is making and pumping, then we have the development of within the umbilical cord, the umbilical vesicles, the arteries, and the vein. But most of what is the nutritional source is really at the molecular level. And so there's really not solid waste that's developing. It's all staying relatively liquid. And it's just a matter of balance of how much glucose, how many, and what types of amino acids the embryo, as it becomes the fetus, is retaining. So what they're excreting is mostly molecular waste in the form of liquid, and that's going into their blood supply because of the filtration units that we have in place starting around week 5 to week 9. Really, there's no excretion of solid waste until after the fetus is born. And woohoo, the very first poop is a big one because it's called the meconium, and that's most babies can't leave the hospital until they've done their first poop. So that's probably way more information that you want that you wanted. But the other thing I'll remind you of is that so it's being moved into the embryonic fetal blood supply, but some of it, meaning the excess glucose and some of those excess amino acids, are actually being excreted, as Dominic just said, into that amniotic fluid. So that's another sort of redundant way for an embryo or a fetus to take on that nutritional supply and not lose it over time. So again, we talked about this at one of our sessions. You can see with ultrasound, you can see the little fetus. It's actually then drinking in the amniotic fluid, and that's all necessary and part of the developmental process. Okay, so I'm going to stop talking now.
Do you have a question, Sammy, if you wanted to unmute?
Yes. So for the local competition, will it be at the same place that I had taken it last year, or different places, or do you know?
So the local competition is the virtual competition, so that will vary for each of you depending on who your nearest local chapter is. I believe that yours, from what I remember, you being in the South, that you still have the same local coordinator, but that will be held virtually. For some of you, you might have your coordinators reaching out and saying that they offer an in-person component. That just means that they're, creating a space for you, that they will help proctor if you prefer to take an examination in person in that way. But it's going to be done in the same format on a computer like it would for anybody else. So that'll be kind of region specific. And so how do you find out where the local chapter is? So all that information will be sent out to you guys closer to the date of the local competition. Once we've closed registration, then we'll finalize your contact information and send it to your local chapter. You don't need to do anything on your end. And they should contact you with the specific Zoom link that you will need to sort of log in and check in prior to starting the exam. So all those instructions will be sent out to you. I know that you are receiving emails if you're at this session. Then we know that you're getting sessions. And just a quick note, if anyone is watching these after the fact and you're not getting anything in your email, just note that there might be an issue with the email address that you provided, and please contact us if you hadn't gotten any of these tutoring emails. Because we'll need to contact you some way to get you that Zoom link.
But I was just wondering because our last year they told us to go to like our local hospital and do it on the computer, so I didn't know if that was okay.
Yeah, they'll be in contact with you. Yeah, and some it depends on the local chapter whether that's something they offer or not. But the exam is the same for everybody, regardless of whether they offer that in-person component.
Yes, ma'am. Thank you.
Yeah, absolutely. All right, if that's all everyone's got, I can't close it better than Dr. Peterson did. So I hope everyone has an amazing holiday, and we'll see you in the new year. Bye, everyone.